All right, welcome to the deep dive. Today, we're going to be doing something a little bit different.
Yeah, should be fun.
Yeah, so think of this as like a tailored exploration just for you.
Okay.
So, you're a philosopher, right? And you're stepping into this world of molecular genetics.
All right.
We've got this like really in-depth entry from the Stanford Encyclopedia of Philosophy.
Okay, great source.
It is. It is. And our mission to like totally dissect molecular genetics. We're going to start with the basics, like what it is, how it works, but then we're going to like dive deep into the philosophical stuff it brings up.
Right, the juicy stuff.
Exactly. So think classical versus molecular genetics. What even is a gene, really? How do those genes do their thing? And why are we so obsessed with them in research?
Yeah, all good questions.
Big questions. It's like a crash course on how molecular biology tackles like the biggest philosophical questions: reductionism, the nature of concepts, information, causality, like all the good stuff.
Sounds ambitious.
Let's do it.
All right, let's unpack this. So first off, molecular genetics itself, well, that's not so simple.
Oh, really?
Yeah, it's got these like different layers of meaning. On one hand, it's this big theory about genes, like they're calling the shots, making those polypeptides, orchestrating life.
Right, like the puppet master.
Yeah, exactly. But then there's the more, I guess, down-to-earth theory, focusing on how genes actually get expressed. All the nuts and bolts, the regulation.
Okay, so like the mechanics of it all.
Exactly. And then it's also the name for this whole toolbox, all the techniques and stuff we use in biomedicine to actually mess with DNA.
Wow, so it's a theory, a set of tools, and a whole approach to research. That's a lot packed into one term.
It is. But for our philosophical journey here, the key thing is that basically all genetics nowadays is molecular.
Makes sense.
Yeah. So when we talk about genetic theory today, we're pretty much in the molecular world.
Okay, I got it.
So as our Stanford entry points out, we're zeroing in on the theory here, and especially how it links up with classical genetics and, the whole idea of what genes are supposed to be doing.
Right. Okay, so before we get too deep into the molecular stuff, let's take a step back to those classical genetics roots.
Good idea.
Think way back, early 20th century, Mendel, T.H. Morgan, all those pioneers.
Yeah, the groundwork.
They laid the foundation, right,
Yeah.
for how traits are passed down. And they came up with this crucial difference between an organism's genotype, its genetic blueprint, and its phenotype, what you actually see.
Yeah. Genotype versus phenotype, classic distinction.
Totally. And the big idea, genotype mixed with environment causes phenotype.
Right. Nature, nurture.
Exactly. They explain heredity by tracking how different versions of these things called genes, these like abstract units, were passed down through generations.
Right, like these hidden factors determining traits.
Yeah, precisely. Like a good example is the eye color of the fruit fly, Rhedocephala. You've got those purple eyes, which are recessive, and the normal red ones dominant.
Yeah, Drosophila, the workhorse of genetics.
Right. So classical geneticists would do this cross, say a red-eyed female and a purple-eyed male.
Okay, classic breeding experiment.
And all the kids, first generation, they've got red eyes.
Because red's dominant.
Exactly. And they explain this by saying, well, each kid gets a copy of the eye color gene from each parent. Mom's got 2 reds, dad's got 2 purples, so each kid gets 1 red, 1 purple.
Right. And since red is dominant...
Red eyes it is. And here's where it gets super interesting, philosophically speaking. Right?
Oh, yeah?
They figured all this out, this whole heredity thing, predicting how traits get passed down without even knowing what a gene was made of or how it actually worked at the molecular level.
Yeah, it's pretty amazing when you think about it.
Right. They just knew these genes, whatever they were, these different versions, alleles, they were the things making the difference in the traits.
Yeah, like placeholders for something they didn't fully understand.
Exactly. The Stanford entry calls this the difference principle, like differences in a classical gene lead to differences in the phenotype. It was all about tracking these abstract units and how they were linked to what you could actually see.
It was brilliant, really.
Totally. But then molecular genetics comes along and boom, concrete answers to all those big questions classical genetics couldn't quite answer.
Like filling in the gaps.
Exactly. So what is a gene, really, in material terms?
Well, molecularly, a gene is a sequence of nucleotides in a DNA molecule. That famous double helix with the GC and TA base pairs.
Right, the iconic DNA structure.
And this is important, not just any chunk of DNA is a gene. It's got to have a function, a purpose.
Okay, so it's not just about the sequence itself, it's about what that sequence does.
Exactly. Think of DNA like a huge book, and genes are the sentences or paragraphs that actually have a meaning, instructions.
That's a great analogy. Okay, so we've got our molecular definition of a gene, but how does this genetic information actually get passed down? Classical genetics couldn't really explain the nuts and bolts of that process.
Yeah, that was a big mystery back then. But the structure of DNA provided a really elegant solution. So gene replication happens when those two intertwined strands of DNA, they unwind.
Okay.
And each of those original strands becomes a template, a model, for building a brand new complementary strand. It follows those base pairing rules, so A always pairs with T and G with C.
Right, like a mirror image.
Exactly. So you end up with two identical DNA molecules, both with the same genes as the original.
It's like a super precise copying machine.
Right. High-fidelity copying, ensuring that genetic information is passed on accurately.
Amazing. Okay, now for the big question from a biology standpoint. What do genes do? We know classical genetics could only observe their effects on the phenotype. What does molecular genetics tell us about the how?
Okay, so the main thing genes do, they act like blueprints for making RNA molecules.
RNA, like DNA's cousin.
Sort of. So the sequence of nucleotides in the RNA is copied directly from the DNA sequence of the gene. This process is called transcription.
Okay, so it's like transcribing a text from one language to another.
Yeah, good way to think about it. And these RNA molecules, they've got all sorts of jobs in the cell. But a really important one is acting as messengers. They carry genetic information from the DNA to the ribosomes, which are like the protein factories of the cell.
So they're like the delivery service for genetic instructions.
Exactly. These messenger RNAs, or mRNAs, they act as templates themselves for making polypeptide chains. Those are chains of amino acids that fold up into functional proteins. And proteins, as you know, they do pretty much everything in the cell.
Yeah, proteins are like the workhorses of the cell, right?
Totally. And the specific sequence of amino acids in that polypeptide, it's determined by groups of 3 nucleotides in the mRNA called codons.
Okay, triplets of nucleotides.
Right. And those codons, they match up with specific amino acids. So the gene, that original DNA sequence, is basically dictating the blueprint for building that protein.
Wow, so it's this whole chain of command, from DNA to RNA to protein.
Exactly.
And what happens if there's a change in the gene, a mutation in that DNA sequence? How does that affect the trait we actually see, the phenotype?
Well, that's where this whole chain reaction comes in.
Hmm.
So if the DNA sequence changes, that can change the mRNA sequence too.
Okay.
And if the mRNA sequence changes, well, that could mean a different amino acid gets put into the protein.
Got it.
And even changing just one amino acid can totally change how the protein folds up, how it works.
So a tiny change in the DNA can have a big impact on the protein's function.
Exactly.
Hmm.
And if the protein isn't working right, well, that can show up as the difference in the organism's traits. Maybe a change in eye color, for example, if the protein involved in making the pigment is affected.
Right, because that protein's not doing its job properly anymore.
Exactly.
This all paints a pretty, I guess, neat picture of how genes influence traits. It feels kind of reductionistic, right?
Yeah, it can seem that way. But the Stanford entry makes this cool distinction between a basic theory and a fundamental theory of molecular genetics. And that gets to some really interesting philosophical points.
Okay, tell me more about this distinction. What's the philosophical difference?
So the basic theory is what we've been talking about. Genes code for RNA, which codes for proteins.
Right, the nuts and bolts.
Exactly. But the fundamental theory goes way bigger. It says that genes and DNA, they're in charge of everything, all life processes.
Wow, that's a bold claim.
It is. They're seen as the source of all the information, the complete blueprint, the detailed program for how an organism develops, functions, even behaves.
So it's like genes are the masterminds behind everything.
Right, which raises some big philosophical questions about determinism and just how far genetic explanations can really go.
Yeah, huge implications. Okay, so this distinction sets up this massive philosophical debate. Has classical genetics been reduced to molecular genetics? Can it even be? It gets right to the heart of reductionism in science.
Exactly. Philosophers of science, especially those into Nagel's model of theoretical reduction, were all over this question. Can you take the higher-level science of classical genetics and reduce it down to the lower-level molecular stuff?
Right, break it down to the more fundamental level.
And Nagel had these 2 criteria for a successful reduction. First, you got to be able to derive the laws. Like the laws of classical genetics should logically follow from the laws of molecular genetics.
Okay, makes sense. You should be able to build the higher level stuff from the lower level foundations.
Right. And second, you need connectability of terms. Like the key terms in classical genetics should be definable connected to the molecular terms.
So you should be able to translate the language of one theory into the language of the other.
Exactly. And if you could do both those things, well, philosophically, it means you've got this unity. The lower level theory is more fundamental, explains more.
Okay, I'm following. So did philosophers think this reduction was actually happening with genetics?
Some did early on. Like Schaffner argued it was happening, classical to molecular.
Okay.
But this view, it ran into some serious roadblocks.
What kind of roadblocks?
Well, philosophers like Hull, they developed this anti-reductionist consensus that became pretty influential.
So they were saying, no, classical genetics can't be reduced to molecular genetics.
Yeah, they argued it wasn't happening and probably couldn't happen, at least not according to Nagel's rules.
Interesting. What were their arguments?
They had two main objections: the unconnectability objection and the gory details objection.
Okay, those sound pretty serious. Let's break them down. First, unconnectability. What was their point?
The core argument, especially from Rosenberg, was that those basic terms in classical genetics, like the gene for eye color, you can't just neatly redefine them at the molecular level.
What do you mean?
Well, think about it. We've got this Mendelian trait, purple eye color in fruit flies, right?
Right.
But the molecular stuff behind it, the genes and the biochemical pathways making that pigment, it's super complex. It's not one gene, one trait. It's a tangled web, lots of genes interacting, and one gene can affect lots of different traits.
Right, it's not a simple one-to-one mapping.
Exactly. So you
Yeah.
can't just point to a bit of DNA and say, that's the gene for purple eyes.
Makes sense. It's more like a network with lots of players involved.
Right. So they argued that trying to force this one-to-one mapping between the high-level function, like eye color,
Yeah.
and the low-level molecular mechanisms, it's just not going to work in biology. It's too complex.
Okay, so that's the unconnectability objection. What about those gory details?
All right. So this one, mainly from Kitcher, he said, even if you could theoretically derive some principles of classical genetics from the molecular stuff, it wouldn't actually give you a better explanation.
Really? Why not?
Well, Kitcher saw classical genetics as being about how traits get transmitted across generations.
Yeah.
And he said the best way to explain that is at the level of chromosomes, how they behave during cell division, meiosis.
Okay, so focusing on those larger structures.
Right. But if you try to explain it all at the molecular level, you get bogged down in all these tiny details, the molecular interactions.
Oh, so it's like getting lost in the weeds.
Exactly. It's too much information, and it's often super specific to each situation. You lose sight of the bigger picture.
I see. So the molecular details don't necessarily help you understand the basic patterns of inheritance?
Right. And it might even obscure them with unnecessary complexity.
So sometimes a simpler explanation is better.
Exactly. It's about choosing the right level of explanation for what you're trying to understand.
That makes sense. So you're saying classical genetics explains inheritance with these relatively simple chromosome movements, and molecular genetics would just give us this super complicated picture of all the molecules involved without really adding much understanding.
That's the gist of it. The anti-reductionists saw biology as this layered system, each level with its own theories and language.
Okay, different levels of explanation for different levels of organization.
Right. Classical genetics had heredity, transmission at the chromosome level. Molecular genetics dealt with gene expression and regulation at the molecular level. They argued you can't just mash them together.
Interesting. So they wanted to keep these levels separate to preserve the explanatory power of each.
Exactly. But of course, this anti-reductionist consensus didn't go unchallenged.
Of course not. There's always another side to the story. So what were the counter-arguments? How did people defend reduction?
Well, critics like Sarkar and Waters, they argued that the anti-reductionists were oversimplifying things.
In what way?
Like they said, the unconnectability objection assumed classical geneticists were naive, that they thought genes and traits had this one-to-one relationship.
Okay, so a simplistic view of classical genetics.
Yeah. But they pointed out that even early geneticists, people like Sturtevant, they knew it was more complicated than that. They knew multiple genes could be involved in one trait.
Really? So they were aware of the complexity even back then.
Yeah. So recognizing complexity wasn't something new that molecular biology brought in. It was already there in classical genetics, just at a more abstract level.
Okay, that changes things. So if they understood it was complex, how did they still manage to build a successful theory of heredity?
Well, that comes back to the difference principle we talked about earlier. Classical genetics, it focused on how differences in genes caused differences in traits, even if they didn't know the molecular details.
Right, they were tracking those differences as the key factor.
And The reductionists argue those differences, those heritable factors that classical geneticists identified, we can now map them to specific differences in DNA sequences, thanks to molecular genetics.
Oh, interesting. So even if you can't map the genes themselves perfectly, you can still map the differences between them at the molecular level?
Exactly. So the connectability between classical and molecular genetics, it might not be totally impossible, just more nuanced than a simple one-to-one mapping.
Okay, that's a good point. What about the gory details objection? How did the reductionists respond to that?
They basically said that the idea of cytological explanations being simple and molecular explanations being super complex, it's not always true.
Really? Can you give an example?
Yeah, like Kitcher used meiosis as an example, right? How chromosomes separate during cell division.
Right.
But the reductionists pointed out that the molecular mechanisms behind meiosis, how those chromosomes pair up and separate correctly, they're actually very similar across lots of different organisms.
Oh, so there's actually a lot of uniformity at the molecular level, too.
Exactly. And on the flip side, dominance, which classical genetics treated as one thing, it turns out to have tons of different molecular mechanisms.
So what looked simple at the classical level is actually really diverse at the molecular level.
Right. So saying that the classical explanation is better because it's more unified, well, it's not always that straightforward.
That makes sense. It sounds like the philosophical debate about reduction in genetics, it got a lot more sophisticated over time.
Yeah, definitely. It moved beyond that strict Nagel model of reduction. Philosophers like Wimsatt, Kitcher himself later on, Sarkar, Weber, they explored different ways of thinking about how theories at different levels relate to each other.
So they were looking at things like how good the explanations were, how different levels of inquiry could work together.
Exactly. Like Wimsatt, he said even trying to do reduction is valuable, even if you don't achieve a perfect reduction.
Because it forces you to think more deeply about both theories.
Right. And Vance, he argued that we basically have one big science of genetics now, where classical and molecular methods are all mixed together.
So it's not about picking one or the other, it's about using the best tools for the job.
Yeah. And Waters, he emphasized that genetics is an investigative science. The methods and explanations are all tangled up.
Okay, so the relationship between classical and molecular genetics, it's a lot more dynamic and interconnected than just a simple reduction from one to the other.
Definitely.
This takes us to our next big philosophical question about molecular genetics. What is a gene, now that we have all this molecular detail?
Yeah, the gene. It seems so basic, right?
Yeah, like the building block of heredity.
But it turns out to be a real headache for philosophers of science.
Really? Why is that?
Well, there's this thing called gene skepticism, and it's basically saying that gene is such a vague and fuzzy term now, it might not even have a real definition at the molecular level.
Oh, wow. So it's not as clear-cut as we might think.
Not at all. Gene skeptics like Burian, Porton, Kitcher, they point out how many different things we call genes in molecular biology.
Like what?
Well, there's the bits that code for proteins, but also the regulatory regions controlling those genes, parts of genes, whole transcription units, split genes with introns and exons.
Wait, slow down. What are introns and exons again?
Oh, right. So in a lot of organisms, genes aren't just continuous stretches of DNA. They're broken up into these segments. The exons are the parts that actually get used to make the protein, and the introns are like spacers in between that get cut out.
Okay, so it's like the gene is edited before it's used to make the protein.
Yeah, exactly. So even just defining what counts as the gene in that case gets tricky. And then there's overlapping genes where the same stretch of DNA can be part of two different genes.
Wow, so the picture gets really messy.
It does. And Kitcher, he famously said, a gene is whatever a competent biologist chooses to call a gene.
Ouch. That's a pretty damning statement. So he's saying there's no real definition, it's just whatever biologists decide it is.
Pretty much. And that raises a big philosophical question, right? If this central concept in biology doesn't have a solid definition, does that mean it's meaningless? Does it undermine all of genetics?
Right, that's a big worry. So is that it? Is the gene concept just broken?
Well, not necessarily. Some philosophers argue that there are coherent ways to define a gene, even if they're more complex than the simple textbook definitions.
Okay, I'm listening. What are these more nuanced definitions?
For example, Waters talks about the classical gene concept and the molecular gene concept.
Okay, two different ways of looking at it. What's the classical concept?
It focuses on genes as functional units, things that make a difference in the phenotype.
Right, back to that difference principle?
Exactly. So it's still relevant when biologists are trying to find genes based on their effects. Like if they see a difference in a trait, they're looking for the gene that causes that difference.
So the molecular details of the gene aren't as important as its overall function.
Right. The focus is on what it does, not what it's made of.
Okay, and what about the molecular gene concept? How does that define a gene?
Waters defines it as a specific DNA unit that determines a linear sequence in a particular product.
That sounds pretty abstract. Can you break it down a bit?
Sure. He's basically saying that a gene is defined by four things: the gene itself, the DNA sequence,
Yeah.
the product it makes, like RNA or a protein, the linear sequence in that product, the order of nucleotides or amino acids, and the cellular context, where and when it's being made.
Okay, so it's all about the relationship between those four things.
Exactly. And this definition helps clear up some of the confusion raised by the gene skeptics. Like, whether introns are part of a gene depends on what product you're talking about. If you're talking about the initial RNA transcript, then introns are included. But if you're talking about the final protein, they're not.
Ah, so the definition depends on what you're focusing on.
Right. It's context dependent.
That's clever. What other approaches have philosophers taken to define the gene?
Well, Moss, he distinguishes between genes P and genes D. Genes P are defined by their phenotypic effects, what they do. Genes D are defined by their DNA sequence, what they are.
So that's like two sides of the same coin.
Yeah, exactly. It highlights the different ways we use the term gene.
Okay, what else?
Weber, he looked at the history of the gene concept, how its meaning has changed over time.
Oh, a historical perspective.
Yeah, he talked about how new technologies and experimental methods have changed how we think about genes, and he called this floating references, like the meaning of gene shifts depending on how we're studying it.
So it's not a fixed concept, it evolves with our understanding.
Exactly.
Makes sense. Anything else?
Yeah, Newman Held, she proposed this process molecular gene concept, which is really important in developmental biology.
Okay, what's that about?
It sees a gene not just as a static piece of DNA, but as a whole process involving DNA and other stuff in the cell to make a protein.
So it's about the gene in action, not just its structure.
Right. It emphasizes the dynamic interactions involved in gene expression.
That's cool. Are there any other approaches to defining the gene?
Some philosophers, like Stotts and Griffiths, they decided to ask the scientists themselves. They did surveys to see how biologists actually use the term gene in their research.
Oh, so they went straight to the source.
Yeah. They wanted to see if the philosophical definitions matched up with how scientists actually think about genes.
That's a really interesting approach.
It is. And then there's Keller. She has this more pragmatic view. She says maybe the vagueness of gene is actually a good thing.
Wait, really? Why would vagueness be good?
Well, she argues that it allows for flexibility in communication across different areas of biology.
Okay, so it's like a common language, even if it's not perfectly precise.
Right. And she sees genes as these manipulable handles that we can use to study biological processes.
So it's more about their practical utility than their strict definition.
Exactly.
That makes sense. Okay, so we've explored this whole debate about what a gene is. Now let's move on to our next question. What do genes and DNA actually do? We talked about that fundamental theory earlier, the idea that genes control everything. But there's clearly more to it than that.
Yeah, that fundamental theory, it's been super influential. This idea that genes are the source of all the information for how an organism develops and functions.
Right, like the genome is the master plan.
Exactly. But philosophers have raised a lot of objections to this idea of genes as the sole carriers of information.
Yeah, the whole concept of information in genetics seems to be a bit controversial. What are some of the criticisms?
Well, for starters, Keller has argued that the analogy of a program or blueprint is way too simplistic. It doesn't capture the complexity of development, how genes actually interact with the environment and other factors.
Right, it's not just a set of instructions, it's a dynamic process.
Exactly. And Oyama, she's famous for arguing that information doesn't just come from genes, it comes from all sorts of sources during development.
Like the cell, the environment, the organism's history.
Right. And she says information isn't static, it's constantly being created and interpreted during the organism's life cycle.
Okay, so it's not like a pre-programmed set of instructions, it's more like a continuous flow of information.
Yeah, much more dynamic view.
What other criticisms are there of this informational view of genes?
Griffiths, he's pointed out problems with both causal and teleosemantic theories of information as they apply to genes.
Okay, can you remind me what those are again?
Sure. Causal theories basically say that information is transferred when one thing causes another thing to change.
Okay, so it's about cause and effect.
Right. But Griffiths argues that if you apply that to genes, then the environment could also be seen as providing information because it influences how genes are expressed.
That makes sense. The environment plays a huge role in development, too.
Exactly. And teleosemantic theories, they tried to define information in terms of function, what something is supposed to do.
Okay, so it's about purpose.
Right. But Griffiths points out that natural selection acts on the whole organism, not just on the genes themselves.
So the function of a gene can't be understood in isolation from the rest of the organism.
Exactly. And Weber, he's criticized the common computer analogy for genetic information, saying it implies that genes have intentions or goals.
Right, like they're consciously directing development.
Yeah, which is misleading. So all these critiques are pushing us away from this idea of genes as the sole, all-knowing sources of information, and toward a more interactive, dynamic view of development.
Okay, so if this simple informational view of genes is so problematic, what are some alternative ways of thinking about what genes and DNA do?
Well, Jablonka, she suggests a broader biological concept of information, where it's all about how a receiver reacts to a signal.
So it's not just about the sender, it's about how the receiver interprets the message.
Exactly. And she argues that genes are just one of many sources of information in this sense.
Okay, so genes aren't special in that regard, they're just one part of the system.
Right. And she makes a distinction between DNA being about the cell, like describing its potential, versus being for the cell, like giving it direct instructions.
Interesting. So the same DNA sequence can have different meanings depending on how it's interpreted.
Exactly.
Yeah.
And Waters, he advocates for a more straightforward causal approach.
Okay, what does that mean?
He says we should just describe what genes actually do, how they cause things to happen, without getting caught up in this idea of information.
So focusing on their concrete effects.
Right. He uses these concepts of actual difference-making and causal specificity.
What do those mean?
So actual difference-making means that a specific gene difference actually causes a difference in a trait.
Okay, so it's about showing a clear link between gene and trait.
Exactly. And causal specificity means that a specific gene difference leads to a specific difference in the RNA or protein it makes.
So it's about showing that genes have precise effects on their molecular products.
Right. And Waters argues that genes are best understood as these specific causal agents that make a difference in the molecular world.
Okay, so it's a more grounded, mechanistic view of what genes do.
Exactly. And then there's Weber and Rosenberg. They've shown how some developmental processes can be explained in terms of how proteins regulate the expression of genes.
So it's a network of interactions where proteins can turn genes on or off.
Right. And this kind of explanation doesn't need to rely on this idea of a pre-programmed genetic program. It's about understanding the specific causal interactions happening in the cell.
Okay, so it's a more dynamic and interactive view of development.
Yeah, much more so.
This brings us to our final question, and it's a big one. If this idea of genes as the master controllers of life is so philosophically shaky, why is so much research still so focused on genes and DNA?
Yeah, that's the million-dollar question.
It's like we can't escape the gene-centric view, even though we know it's not the whole story.
Right, and it's tempting to just say, well, it's because of genetic determinism, this belief that genes determine everything about us.
Yeah, like genes are destiny.
But the Stanford entry offers a more nuanced explanation. And it goes back to what Keller said about the practical usefulness of genes.
Okay, how so?
Waters argues that genes are really useful tools for studying and manipulating biological systems.
In what way?
Well, they're like these reliable difference makers.
Okay, remind me what that means.
It means that if you change a gene, you can often see a predictable change in a trait.
Right, so they're like these controllable variables that you can use to experiment with.
Exactly. They're like handles that you can grab onto to study how things work. And that practical utility, it's a big reason why gene-centric research is so popular.
So it's not necessarily about a deep belief in genetic determinism. It's about using genes as tools to understand biology.